All right, so the final thing we're going to talk about for this module is sexual reproduction and the cellular division process that governs the generation of our gametes, so our sperm and our eggs. That cellular division process is known as meiosis. And it is what leads to the generation of the gametes and the unique combination of alleles that exist in our gametes. And then that unique combination is also even more randomized due to the process of fertilization. And so we're going to go in all that detail in this video of the module. So we said sexual reproduction is the production of offspring where there's a unique combination of genes that comes from the parents. And we get one set of our chromosomes from one parent and another set of our chromosomes from the other parent. And the specific combination of chromosomes depends on the process of meiosis. And it also depends on the process of fertilization. So fertilization is random. So, it's random in what sperm fertilizes the egg and which egg gets fertilized at a given point in time, and each egg is different. And so that whole fertilization process, is random and is random in determining what sets of chromosomes our offspring will have. So and meiosis is also the reason why we see such a variety in our offspring as well. It's why we see traits appear in our children that don't appear in the parents, and it's also why our children favor the parents a lot in terms of their traits and behaviors. And so again, that all comes from the fact that meiosis splits and determines what alleles and what chromosomes will be put into the gametes before fertilization happens. And now we've talked about chromosomes in mitosis. We talked about sister chromatids. Now we're going to complicate things a little bit, and we're going to add in the term homologous chromosomes. Okay. So we mentioned that different species have different numbers and types of chromosomes. And if you remember, humans have 46 chromosomes, 23 pairs, and those pairs of chromosomes are known as homologous chromosomes. So each of the 23 pairs have the same DNA and gene pattern within, not the same DNA, but the same gene pattern within that chromosome. And we'll look at a picture of that in the next slide. And a somatic cell of humans is going to have and contain those 46 chromosomes. Now, if we take a cell and we stop it during the division process, we can then produce what's a karyotype. So we take a cell that's dividing, stop it during that process. We break open the cell, and then a researcher can go in and stain those chromosomes with dyes. Then they can rearrange the chromosomes using a microscope and take a picture of all of those chromosomes, and that produces what's called a karyotype. And so here's the karyotype of a male human, and we know it's a male because it has an X and a Y chromosome. A female would have an X and an X. And you can see pairs 1, 2, 3, all the way to 23. So the sex chromosomes are the 23rd pair. So you have 22 autosomes, which are found, which don't determine sex. And then your sex chromosomes determine the gender. So that's the 23rd pair. And what you can notice with these pairs of chromosomes is that the banding pattern, which each band pretty much represents a gene. And so you can see there's a gene here that's pink, this orange one, and this green one. They match in size. And so that banding pattern is the same because those chromosomes have the same genes, right? And so let's say, this first chromosome has eye color. gene at the top. And so you can have different alleles for eye color on the, say, paternal chromosome, which is maybe the left one, and the maternal, which is maybe the right one, right? And so these are homologous chromosomes because they have the same genes and they're the same chromosome type, okay? And it's important to know the homologous chromosomes versus the sister chromatids because the homologous chromosomes separate at a different time during the meiosis division process, and this leads to the differences between, or one of the big differences between mitosis and meiosis. So again, those homologous chromosomes really resemble each other in terms of length and where that centromere is positioned. So basically where the sister chromatids are joined together. And they also carry genes controlling the same characteristics, okay? And like I had mentioned, with humans, we have 46 chromosomes. 22 pairs are called autosomes, and then our 23rd pair is called our sex chromosomes. And that's either going to be an X or a Y, and so you can have a combination of XX or XY, depending on if you're male and female. And now with the life cycle of a multicellular organism, we have these generation to generation stages. So we have fertilization to development of the embryo to the development of adults that are reproductively active and can then develop their own gametes and have their own children. And so with that process, each parent has two sets of chromosomes, and only one chromosome from each pair is inherited from each parent. And so that's the key factor in the life cycle of any species that reproduces sexually. And so here we have a figure that's kind of showing the generation of the human life cycle. You have your adults that produce their gametes, and then their gametes undergo fertilization that generates the zygote that develops into the embryo and then develops into the fetus and is born as a human and then develops into an adult that then undergoes meiosis and generates their own gametes and their sperm and eggs. Now, there's also some notation here that I want to point out, and that's haploid versus diploid. So haploid means that the cells contain two copies of each chromosome. So haploid individuals in humans have 46 chromosomes, or haploid cells have 46 chromosomes, and we denote, diploid. So diploid has two copies of the chromosomes. I said that wrong. So your diploid cells have two copies of chromosomes in each cell. And so the diploid cells, so the zygote up to adults, will have two copies of each chromosome, and that's denoted by the 2N that's shown here. So basically N being your chromosome and 2N saying you have two of those chromosomes. So you can see that in the legend here as well. So 2N. The only portion of the life cycle for humans that is haploid are the gametes, the sperm and the egg cell. That haploid means that they only have one copy of each chromosome. So they only have 23 total chromosomes. And that's because when fertilization happens, those chromosomes combine in that diploid individual to produce a 2N cell. And so we undergo a majority of our life having haploid cells, and then we have diploid cells, and then we produce haploid gametes that will undergo fertilization and develop into a diploid individual. So like I said, humans are diploid because all of our body cells contains pairs of homologous chromosomes. And so that haploid cell only has one copy of each of the 23 chromosomes. And so within that human life cycle, your haploid sperm fuses with the haploid egg. That's fertilization. And then the resulting fertilized egg is the zygote, and that zygote is diploid. Okay. The egg and sperm is produced through the process of meiosis. And meiosis keeps the chromosome number from doubling every generation. So if our sperm and egg had 46 copies of chromosomes, then the remaining zygote would have 92 chromosomes. And then if their sperm and eggs had 92 chromosomes, then the resulting zygote would double the numbers of chromosomes again. And so meiosis reduces the number so that our chromosomes don't double every generation. And it also is important because we have certain protein expression, certain gene expression. And if you have more than the needed chromosomes or less than the needed chromosomes, you can end up with disorders, and you can end up with proteins not functioning correctly, which leads to those disorders. And a lot of times not having the correct amount of chromosomes can be fatal, and the zygote or the embryo doesn't even make it to birth, or they typically die at a very young age because of the resulting disease. And so it's very important for that process of meiosis to go perfectly so that the number of chromosomes in the gametes are the appropriate number. And we'll talk about how errors can happen in meiosis towards the end of this video. And so meiosis, like we said, halves the number of those original chromosome numbers. And so for humans, we go from 46 in our parent cell to 23 chromosomes in our daughter cells. And meiosis does this through the process of two individual division phases. Okay, so the first division phase goes from duplicated homologous chromosomes. So homologous chromosomes that have sister chromatids, so that means they're duplicated. And those homologous chromosomes separate. So one copy of that homologous chromosome will go into one daughter cell, and the other copy will go into the second daughter cell. Okay, and these sister chromatids will now undergo another division phase to where we separate those sister chromatids just like we did in mitosis. So they'll separate, and you end up with one copy of each chromosome in the daughter cells. If you start with one cell, you end up with 4 daughter cells through the process of meiosis. So one cell divides, gives you 2, and then those 2 daughter cells divide again, and that gives you a total of 4 daughter cells. Before meiosis happens, the cell will go through the cell cycle. It goes through interphase, just like the cells in mitosis, and it goes through the S phase. So the chromosomes will duplicate. And so here you have that non-duplicated chromosomes, and you have those homologous chromosomes. So you have the paternal, which is the blue, and the maternal, which is the red. And then they'll be duplicated like we see before we start the process of meiosis. Okay. And so as you probably noticed in that figure, there are a couple of differences between meiosis and mitosis. The first big difference is that mitosis only occurs in gamete cells, whereas meiosis only occurs in gamete cells, whereas mitosis occurs in all of our somatic cells, so all of the other cells throughout the body. The second big difference is that the number of chromosomes in our daughter cells in meiosis is cut in half. So you go from 46 to 23 in humans. Another difference between meiosis and mitosis is the fact that genetic material can be exchanged between the pieces of homologous chromosomes. And this process of exchange is called crossing over. And it only can occur during the first prophase of meiosis. after the first prophase of meiosis happens, crossing over cannot occur. So it only can occur during that particular step. And so those are three real big differences that I want to make sure you're aware of whenever we're comparing mitosis to meiosis. So here we have a figure showing all of the stages for meiosis. So here you have your normal gamete cell that has the nuclear envelope, the uncondensed chromosomes, the centromeres, and the cells in interphase, and it's just kind of doing its thing, right? Growing, getting larger, and then in the S phase and interphase, those chromosomes will duplicate. And then after duplication, they continue to grow in the G2 phase, and then they go into the meiosis division process. And then you have two division phases. That first division phase, which is these parts of the figure here, is referred to as meiosis 1. And this is where your homologous chromosomes separate. So meiosis 1 is made up of 4 phases. So prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Yeah, so those are always kind of lumped together. And so the order of the phases is the same way as mitosis, except for meiosis, we call them prophase 1 and prophase 2, depending on which division phase we're in. So with prophase 1, the exact same thing pretty much happens as in mitosis. The nuclear envelope breaks down, your centromeres start developing the spindle fibers and start moving towards the poles of the cell. But the other thing that happens in prophase 1 that doesn't happen in mitosis is the crossing over, okay? And do they show it in this figure? I'm trying to look and see. Yes, they do. So what you can see in this figure, let me show. So right here, you have two sites of crossing over. So basically what happens is, imagine if you have two pieces of string, a blue and a red one, and you cut the ends off of them, and then you take the red piece you cut onto the blue string, and the blue piece you cut onto the red string. That's exactly what happens with crossing over. Is you're taking those chromosome pieces, the cells kind of cutting them off and grafting them onto the opposite one. So they're basically switching places. And you can kind of see that in this figure here. You can see that the blue now has a red tip, and then the red chromosome has a blue tip, okay? That crossing over can only happen with the homologous chromosomes that are close to one another. You see with this pair of homologous chromosomes, there's two that are close together, and then this red one and this blue one's kind of away from each other. Those two that are away from each other on the ends, they can't cross over. It happens with these internal portions of the pair of homologous chromosomes, okay? And so you have two pairs of homologous chromosomes in this figure. One has underwent crossing over, and each of those pairs of homologous chromosomes have the sister chromatid for the paternal and maternal chromosomes, okay? Now, after prophase 1, so after crossing over, after the nuclear envelope has broken down, after the centromeres have started developing the spindle fibers and started moving towards the poles of the cell, your homologous chromosomes will now line up on the metaphase plate as it moves into metaphase 1. This is different from mitosis because remember, mitosis, all of the chromosomes lined up in a line on the metaphase plate, whereas in metaphase 1 for meiosis, the homologous chromosomes line up. Okay, so you can see the pairs. Excuse me, the pairs are lining up along that plate, and so that plate would be kind of like right here. Okay, so you have a homologous chromosome on this side and a homologous chromosome on this side. Now, one interesting fact about this, and we'll talk about it a little more, is that this leads to the process called independent assortment. So notice how in this figure you have a large blue one on one side and a small red on the other side. Notice they didn't line up red, red, and blue, blue. This means you'll get a paternal version of one chromosome to be in this daughter cell and the maternal version of another chromosome to be in that same daughter cell. Whereas if it was blue, blue, and red, red, that daughter cell would contain all paternal or all maternal chromosomes. And that is a random factor that can happen. And it can lead to a lot of variation. If you imagine 23 pairs of homologous chromosomes lining up and kind of flipping, alternating along that metaphase plate, that's a ton of different possibilities and combinations. And so that's also why there's so much variation in terms of genes that we have whenever we pass those genes onto our offspring. So two ways that we've talked about already that leads to increased variation in the genetics of our gametes is crossing over, that's one, and the second is independent assortment, that's two. So crossing over occurs only in prophase one, independent assortment occurs in metaphase one. That's the only two places they can occur. So once those homologous chromosomes have lined up along the metaphase plate, the spindle fibers will grow out and grab onto the centromeres of the homologous chromosomes. And in anaphase one, those spindle fibers will shorten. And as those spindle fibers shorten, the homologous chromosomes will be pulled away from one another and they'll go to their respective poles of the cell. Okay, so now those homologous chromosomes are split. Okay, now you only have one copy of each chromosome in the daughter cells. So after telophase 1 and cytokinesis happens, so your nuclear envelope reforms, you get a cleavage furrow. After that happens and you end up with your two daughter cells, your cells are now no longer diploid. They are now haploid. So two haploid cells will form, but the chromosomes are still replicated. They're still doubled. So the chromosomes are still copied, but they're haploid. There's only one version of each chromosome in each of these two daughter cells. Okay. Now each of these two daughter cells will move on to the second division phase, meiosis 2. And then the phases will start back over. You'll have prophase 2, metaphase 2, anaphase 2, and telophase 2 in cytokinesis. This is almost exactly like mitosis. You have your, in prophase 2, you have your nuclear envelope to break down in both cells, your spindle fibers to start growing again, and your chromosomes are condensed. Metaphase 2, your sister chromatids, so now they're only sister chromatids. They line up along the metaphase plate. And notice you still have one copy of each chromosome. So there's a large chromosome in this cell and a small chromosome, a large in this one and a small. It's just that their alleles are different. So this one has the paternal alleles, this one has the maternal, and vice versa. This is maternal and this is paternal. So still all of the chromosomes, they're just half now. And they're no longer homologous, they're only sister. So sister chromatids line up along the metaphase plate. Spindle fibers attach to the centromere. Anaphase 2, the spindle fibers shorten, and now they pull those sister chromatids apart. Okay, so now once they pull those sister chromatids apart, the chromosomes are no longer doubled. Now the duplicated chromosomes have been separated. And so then you'll have telophase 2 and cytokinesis, which will split each of these two cells, resulting in 4 daughter cells that have one copy of each chromosome. So you'll notice you have one large, one small, one large, one small. It's just that their origin is different from paternal and maternal. Okay. So you want to make sure you can go through my meiosis in that detail. You want to make sure you can look at this figure and you know exactly what's going on, what's happening in these processes. You know where crossing over happens, where independent assortment happens, when the homologous chromosomes are separated, when the sister chromatids are separated, when are the cells diploid, when are the cells haploid. And so that's the important information that you really need to take away from this process of meiosis. All right, so now for comparison, so mitosis, meiosis. So both instances, the chromosomes only duplicate once. There's only one interphase before that division step, even though meiosis undergoes 2 division phases. Mitosis is used primarily for growth, tissue repair, and also asexual reproduction. So for like your single-celled organisms. The offspring of that parent daughter cell, so those of that parent cell, produces 2 genetically identical daughter cells, okay? And they're identical to that parent cell. Meiosis, that's not the case. Neither for sexual reproduction, the products, the 4 daughter cells are genetically unique from each other and also from the parent cell, and they are haploid, they are not diploid. And the cells only have one member of each homologous chromosome pair, and that's going to differ in being either paternal or maternal version of that homologous chromosome. Okay, so here we have a comparison figure kind of showing those differences and also where the important stuff happens. So like crossing over in meiosis 1, separation of homologous chromosomes, you have your sister chromatids in the prophase of mitosis, etc. Right? So this is just a really good summary figure for you all to look at and use to get an idea of those comparison processes. And what's really great about these figures is it shows crossing over and it carries crossing over through the diagram. So you can see the little pieces that have been grafted onto the opposite homologous chromosomes. Also, all the events that occur in meiosis 1 are unique to meiosis. Okay, meiosis 2, like I mentioned, is almost identical to mitosis in that it's separating sister chromatids, but unlike mitosis, it's giving you haploid daughter cells instead of diploid daughter cells. That's the big difference. Okay. So here we have just a review question for you to think about. So take a minute to pause the video and think about this one. So true or false? Both mitosis and meiosis are preceded by chromosome duplication. All right, so the answer to this is true. So both of these cell division phase processes undergo chromosome duplication before they happen. And even though meiosis has 2 division phases, it still only undergoes duplication 1 time. All right, so I've already talked a lot about independent assortment, but I just kind of wanted to give it its own little spotlight just to make sure that you all understand exactly what this is. So independent assortment can happen again in meiosis 1. So whenever the homologous chromosomes line up along the metaphase plate in metaphase 1. And so the offspring that occur through that sexual reproduction process are genetically different. And I've already said, that genetic variation happens through crossing over and independent assortment. And then the third way that happens is just through the random combination of sperm and eggs when fertilization happens. And so independent assortment is one big way that genetic diversity occurs. And so when those homologous chromosomes align on that metaphase 1 plate and align along the metaphase plate during metaphase 1, they have that side-by-side orientation, and they can flip that orientation however that the chromosomes line up, and it's totally random. And each chromosome lines up independently from the other chromosome. And so that's where the independent assortment comes from. And so just because these first homologous chromosomes line up with blue on the left and right on the red does not mean that the second pair of chromosomes have to line up that way either. You could get that orientation, but you also could get an orientation this way. And that completely determines, or I should say, that randomizes the variation within the resulting daughter cells that ends up after the process of meiosis. So for example, possibility one, you carry through the first division phase, you have all blue, all red. Carry through the second division phase, you have all paternal and all maternal chromosomes. However, if we go through possibility two, you have paternal, maternal, and maternal and paternal. It's just that the chromosomes that have those are different, right? And then you can see with combinations C at the bottom, you have a large paternal and a small maternal. With combination D, you have a large maternal and a small paternal. And so that again really affects what alleles are going to be present in the offspring once fertilization happens between those gametes. And like I mentioned, that's 23 different possibilities. And so 2 to the 23rd power. So that's 8 million possible chromosome combinations just at meiosis, just in meiosis 1, so at metaphase 1. You add in, or I should first say that with those 8 million possible chromosome combinations for a man and a woman, you put those randomizing fertilization events, that's about 64 trillion combinations of chromosomes. And then you add in the fact that crossing over can happen. And so that's really like a 1 in a zillion, I don't even know how high the number would be, of combinations that could lead to your specific genetic makeup. So when somebody says you're 1 in a million, so actually, no, I'm more like 1 in a trillion. All right, so process of fertilization. Here's a figure of that, just sperm and egg, egg with a lot of sperm on it. And so whichever one makes it through is completely random, right? And so that determines, again, your final genetic makeup of your offspring. And so we mentioned crossing over, right? So crossing over is that exchange of the segments between the homologous chromosomes, so the non-sister chromatids. And again, that only occurs during prophase 1. And so when you have crossing over, the gametes can basically have chromosomes that are slightly different from the chromosomes that exist in the father and the mother, because that crossing over causes that combination to be different. And we call these recombinant chromosomes because they arose from this process of recombination, which is crossing over. And just to kind of give you a more kind of detailed view, this view is a lot easier to see than in that meiosis figure before, of crossing over. You have your homologous duplicated chromosomes, so your sister chromatids that have paired up. And then those interior sister chromatids will cross over. And so they cross over like shown in this figure, and then they switch that genetic material. And so you end up with the maternal allele on, or the maternal portion of this chromosome on the paternal chromosome itself. And crossing over can occur over the entire length of the chromosome. It's just generally easier to show the ends of the chromosomes crossing over in figures. It's just easier to draw that way. But theoretically, crossing over can occur at any point on those chromosomes. Okay. And so you end up with sister chromatids that resemble this. And then when the final separation happens, this is what is left in the resulting gametes. So because the outside homologous chromosomes did not cross over, so the outside sister chromatids did not cross over, those are not recombinant chromosomes, they're normal. But the ones that are close together did cross over, and that's the recombinant chromosomes. Okay, so one of the last things we're going to talk about are non-disjunctions, so basically errors that can happen during the process of meiosis. when you have errors, they could either happen in meiosis 1 or meiosis 2. In about 10 to 30% of human conceptions, the main reason for pregnancy loss in those conceptions is most likely non-disjunction. So the gametes didn't have the correct number of chromosomes, and so that means that the zygote either had too many or too few chromosomes to develop properly, and so the pregnancy was aborted by the body. The body can tell that a lot of the times. And you can have two episodes of non-disjunctions in meiosis 1 and non-disjunction in meiosis 2. If you have a non-disjunction in meiosis 1, then you get all of the gametes to be abnormal in after that meiosis process has happened, because what that means is one of the homologous pairs do not separate, and so none of the chromosomes for that pair goes on to one of the daughter cells. So you basically will always have one less chromosome in that daughter cell. And so when it divides, you end up with one less. And then the opposite daughter chromosome will always have one more. So all of those resulting cells, resulting gametes, will be abnormal. However, if you have non-disjunction and meiosis 2, then the homologous chromosomes have separated appropriately during the first division phase, but in meiosis 2, one of the sister chromatids doesn't separate, but one of the cells, they could separate. And so you get two gametes being normal, and you get two gametes being abnormal, with one containing more chromosomes and one containing less chromosomes. Okay. And what happens is, if you have fertilization after non-disjunction, say you have an egg that has 3 copies of the chromosomes, or I'm sorry, an extra copy of a chromosome, not 3 copies of the chromosome, but say it has an extra copy of one chromosome, then that means you will have an abnormal zygote if it fertilizes with a normal sperm cell. And so you'll have 47 chromosomes because you have one extra from that non-disjunction event. Now, this is somewhat common. Non-disjunction, somewhat a common thing that happens, and like we said, it leads to 10 to 30% of pregnancy losses. And one of the more common non-disjunctions leads to what's called trisomy 21, or you probably know it better as Down syndrome. And so Down syndrome comes from the fact that the child has one extra copy of chromosome 21. So that's why it's called trisomy. Instead of having 2 copies, they have 3 copies. And it actually affects about 1 out of every 850 children. And it's the most common chromosome abnormality. And that extra copy of that chromosome leads to developmental differences within that person. They typically have characteristic phenotypes, so they'll have typical facial features. They usually have a shorter lifespan compared to normal people, and they also have specific developmental delays and difficulties just because they have that extra chromosome. And it comes down to the fact that because you have that extra chromosome, you're basically making extra proteins that are made by that 21st chromosome. And our bodies don't do well with those extra proteins being made. Our bodies have evolved to develop and grow and work with specific protein quantities being expressed based off the number of genes we have. And so those developmental issues and the phenotype differences, so the way the physical features look, come from that overexpression. generally, and a lot of different other factors that come about from that extra chromosome. A lot of people with proper care, though, with Down syndrome can live to be middle aged or even older. Some of them can be pretty good socially. They can live on their own and they can even hold jobs. It just depends on the severity of it, and also how much help and support they have from, say, family members or other people. And one thing is, we don't really know why the risk of Down syndrome increases as the age of the mother increases, but generally, fetuses of pregnant women that are 35 and older typically have a higher risk of having offspring with Down syndrome, and are recommended to undergo things like parental screenings to check and see if that's a possibility. But then again, people may not care to know, and so they may choose not to have those parental screenings anyways. Some other types of non-disjunctions can actually lead to abnormalities in sex chromosomes, so like your X and your Y chromosomes. So you could get um, offspring that have unusual numbers. So, for example, you could have two X and a Y. You could have an X and two Y, or kind of any combination of that. And it seems to be the case that when individuals have these unusual numbers of sex chromosomes, they seem to really upset the genetic balance less than unusual numbers of autosomes. Um, and so basically what that means is the non-disjunction, or the unusual number of sex chromosomes, doesn't appear to be as significant as the unusual numbers of all of the other 23 autosomes, so chromosomes 1 through 23. And so we have a table that shows the different possibilities for abnormalities in sex chromosomes. So we have, for example, Klinefelter syndrome, which is a male, where they have two X's and a Y. These individuals are typically sterile. They have underdeveloped testes, and they typically have secondary female characteristics. And that makes sense because they have a second X chromosome, which means that they have double the amount of proteins and hormones that's made from the genetic strands of that X chromosome. You also could have an individual that has one X chromosome and two Y. These are normal males, and they're just typically taller than other individuals. You have a female that is triple X chromosomes, so that's a normal female, just slightly taller. And through, I guess, research, they've shown that there could be some learning disabilities associated with it, but I'm not too sure of the evidence that's supporting that, but that seems to be the case in some situations. And then you have this really interesting situation called Turner syndrome, where you have a female with only one X chromosome, so they don't have a Y chromosome. And that's called, again, Turner syndrome. And those individuals typically are sterile, and they have immature sex organs, and so they aren't able to I guess produce gametes, but I'm not real sure about that. And so just real interesting cases in which you can get non-disjunctions of sex chromosomes leading to abnormalities in the numbers of those sex chromosomes and offspring. And just kind of the last thing I want to mention before we end the video for this module is asexual reproduction and kind of the advantages for that versus sexual reproduction. So just kind of thinking about the pros and cons for each. An organism that produces asexually doesn't need to spend energy to make gametes. So our bodies spend energy making sperm and making eggs, and we have to have those reproductive organs. And so our bodies spend energy developing and upkeeping those organs. And we don't have to spend energy or I'm sorry, asexual organisms don't have to spend energy finding a partner. They can just go about their daily life, eating food, gathering resources, and they will reproduce by that asexual method where they either bud off, or you can graft like a plant, or they just divide via what's called binary fission, where the cell will just undergo mitosis and divide. And so that could be also advantageous in terms of evolutionary mechanisms, because if you have an organism that's really good and really suitable for its environment, then you want all of your offspring to be genetically identical so they can do as well in that one particular environment. However, the downside to that is, if something changes in that environment, and say a disease comes in and one cell is susceptible, then all the cells are going to be susceptible because they're all genetically identical. Or if all of a sudden the environment changes and they're not suited for that environment, now all of the individuals aren't suited for the environment and they'll probably go extinct or be lost in that area. So that's kind of the downside for asexual reproduction. Now, for sexual reproduction, you can have evolutionary advantages because you always produce offspring that have that very genetic makeup. There's lots of variability in our populations, because none of our offspring are going to be genetically identical unless they happen to be identical twins. Even if you have fraternal twins, they're still not genetically identical because that was just a case of two sperm fertilizing two eggs. And they just happen to implant and develop at the same time. And so even then, those are varied in their genetic makeup. And so that could be really good if climates change or if we have to adapt, and there could be variation for that to be selected on. And so if they're genetically identical, then there's no variation to select on and be beneficial for, say, changing environmental factors. And it also helps to reduce the incidence of harmful genes really proliferating in the population. And so if you have a harmful gene and you reproduce asexually, then that harmful gene is going to also be in your offspring, and then it'll be in their offspring. And so it can proliferate exponentially at that rate. And so there are pros to cons for both asexual and sexual reproduction. It's important to know those and to be able to talk about the comparison of those two reproduction methods. All right, so that's it for modules 7 and 8. So let me know if you have any questions, if you need any help, and we will see you in the next module.